Thank you, Peter. Don't go anywhere. No. If I can encourage our speakers to come back to the stage, we'll have a slightly shortened Q&A session. Try and get us back on time. And there should be a couple of microphones there available. Okay, Ewan?
Hello, it's Ewan again. I'll try not to ask too many questions. But for both Peter and Zach, the lovely talks, I get a tingling worry about fine-grained geographical structures in these PRSs and GRSs and sophisticated uses of them. You know, in your case, Peter, for looking at this very old question, and Zach, in your case, for this risk prediction. And I know I've talked to both of you about this, but I'd just like to, two things. How paranoid are you about it? And how do you reassure the people who are paranoid? I suspect there are more people in this room who are paranoid about it than I am.
Thank you, Ewan. Yeah, so I think you're asking basically about the external generalizability of the scores derived from UK Biobank. And for any predictor, the key is just take it to other samples and see how well it does. So we've taken this predictor to 3 other samples, separate studies in the United States, and it works almost identically, remarkably, to what we found in the 300,000 people in phase 2. And one of those is a multi-ethnic study of several thousand people who are hospitalized with early heart attack in the United States, and the predictor works just beautifully.
It's a great question, Ewan. So I think to translate it to my talk, is this on yet? So the question is essentially, I think, if people in Cornwall are slightly taller than people in Scotland, and people in Cornwall tend to find their partners there, and similarly in Scotland, then you might also see a pattern of assortative mating, which just reflects mating according to ancestry. Well, we spent the 95% of the time on this project in trying to look at how to deal with population stratification. It's a very good question. We're very well aware of it, and it's the Achilles heel for these studies. We're pretty confident that they were not suffering from that in the conclusions that I've drawn from the traits that I have. But if you're not careful, you'll find evidence, so-called, for assortative mating, which is just pure population stratification. I agree with that.
Okay, thank you, Ewan. Next question over here.
Nick Cross, University of Southampton. As part of the exome sequencing and genome sequencing studies, you're going to pick up individuals with clonal hematopoiesis driven by somatic mutations. And these individuals with an increased risk of developing hematological malignancies. So what's going to be done with that information? Is that going to be fed back to individuals, or what?
Yeah, so that's probably my understanding of it, and certainly UK Biobank team can say more. So we're aware of those types of variants, and some are ones that others have recognized, and we already see that in other exome sequence data sets and can see that here. And there's certainly many important research questions that are going on from that, but this is not something, nor will any other genetic information, I think, at the time, be returned back to these participants, but that's kind of, I think, beyond our applicant
Rory?
comments.
Yeah, maybe I could speak to that. So everybody who participated in UK Biobank did so on the basis of clear consent. There will be no feedback of individual results to the participants. As we've gone through the imaging assessment as well, we've reiterated that point to participants. During the baseline assessment visit, if an incidental finding was noticed, then that would be mentioned to them. Similarly, in the imaging, if during the actual imaging assessment the technician notices something of concern, then that's checked by a radiologist, and about 2% of participants then get feedback from something spotted during the assessment visit. But the consent is to take part in the study originally and reiterated for the imaging is that anything found subsequently will not be fed back.
Thanks, Rory. Next
I could
question
just
over
add
here.
one more thing on this. So I think it's an important area of research that is really, just starting to be looked at in terms of the consequences of those. And this data set will be great to look at that. And we've had some experience looking at our Geisinger data set, which has a much wider age range, so from 30 to centenarians. And so you can detect a lot of these simply by looking at different age strata and their presence there. And we also have, like UK Biobank, a very broad phenotypic catalog, if you will, there. And so just some caution, we have to be quite careful about the associations we make there, because there's a lot of age-associated phenotypes, and causality we ascribe to some of these variants to certain phenotypes versus just a byproduct of age is going to be a very important question.
Okay, thank you, Maris. Do you have another question over here?
Hi, Daniel Chubb, Institute of Cancer Research. All of the really great advantage of the exome sequencing project are also entirely captured by the whole genome sequencing, with the added advantages of evenness of coverage, not due to capture inefficiency in exome sequencing. So why do both, or why continue with the exome sequencing when the whole genome's going to do it all anyway?
Well, I think we've all, talked about this, and we're all kind of complementary approaches, and I think that's in fact the short answer. They're all complementary. I think everyone wants to have the value of 50,000 exomes now, 500,000 exomes by the end of next year. Does anyone want to pass on that opportunity and wait for 5 years to have everyone sequenced? And that challenge is an enormous challenge in terms of kind of scale and infrastructure, and it will add some additional valuable information that Nicole talked about. So I think they're extremely important initiatives. I don't think anyone here would probably disagree with that we ought to carry on with them full steam ahead.
Yes, and to add to that, clonal hematopoiesis is a clear example of, you'll have a better resolution from a 50x or 60x exome than you would do from a 30x genome. So I think there is complementarity, not only, timing is obviously of the essence, but also in terms of the biological information that we can get.
Yeah, another thing I can say, in 10 minutes you can't say much, but thank you to Mark for an extra thank you to John Overton and Jeffrey Reed, who's here, and they have done a really exquisite job in terms of the sequencing and the informatics. The coverage in these exomes is extremely high. So mean coverage is not the best metric when you're talking about exomes. So we'll look at the proportion of targeted bases that have, let's say 20x coverage or so, right? And on average now, in this first, QC report of the 50K, we're talking about 94 to 95% of targeted bases at 20x or higher. That's high. So this is high quality data.
Okay, thank you. I think we probably have time for one more question. This question just here.
Hello, I'm Özlem from Boğaziçi University, Istanbul, Turkey. So I want to ask that there is current genomic data in the UK Biobank, and the exome sequences are added in the current study. So what is the additional information when we compare the genomic data currently for the whole data set and the exome sequencing data?
So I try to give a little bit of a teaser on that, but essentially, and you also heard about it in the whole genome talk, that imputation is a fantastic tool, and it will continue to improve with more sequencing and better reference panels, but it can only go so far. And as you get to a certain allele frequency, imputation accuracy is quite poor. And one way to think about genetics is it's all rare variation. Certainly when you get to this population scale, the common variation plateaus quite quickly. And so the overwhelming majority of all genetic variation you're going to see is going to be rare variation. So the sequencing efforts are filling in an enormous amount by counts and other metrics of variation that doesn't exist, period, or isn't very accurately imputed in the data. So that's what I would say in terms of what
so if you
sequence
think
adds.
a rare variant will be a best call about 10% of the times in an exome sequencing data set, in an imputation data set, but we can recover the completeness of genetic variation down to variants that are carried by a single person from all exome and all genome sequencing data. And I think the other point that I mentioned as part of the DD project, but it's really an important one, is that having a genome-wide distribution of variation really allows you to model extremely accurately pressures, on the genome. So, for instance, selective constraints or mutational constraints, which we know very well are not constant over the genome. So our ability to really understand what is a non-normal pattern in a genomic region, for instance, a disease-associated variant, really critically depends on our ability to model genetic variation throughout the entire genome. So having that background information that tells you, for instance, in that region mutation rates are higher or lower than average, also informs how you interpret the phenotypic effect of this variant. And so for this reason, these data sets are all highly complementary. They all give you a different part of the picture.
Okay, thank you. I think we probably need to bring the Q&A session to a close. What I would suggest is if you have a burning question, then please try to collar our speakers after this session. So we'll draw this to a close, and I'd like to thank our speakers for their presentations.